Okay, let's dive right into this explainer. If you've ever looked at the absolute sheer variety of life on Earth, from the microscopic bacteria living on your skin to those massive fungi out in the forest, and wondered how on Earth scientists keep it all organized, well, you are in the exact right place. Today, we are taking the incredibly complex world of biological classification and, quite frankly, just breaking it down. We're going to use simple, easy-to-memorize tricks and bullet points to help you instantly understand exactly how life is sorted.
But first, we need a little context. Early attempts to sort life were, well, they were pretty basic. Take Aristotle's morphological trick. It was super simple. He literally just looked at plants and said, okay, are you a tree, a shrub, or an herb? And for animals, his big dividing line was simply whether they had red blood or not. Later on, Linnaeus came along with his two-kingdom trick, sorting everything neatly into either Plantae or Animalia. And that worked for a while. But ultimately, these systems failed. Why? Because they couldn't cleanly handle microscopic misfits, they clumped together things with entirely different cellular structures, and they totally ignored complex organisms like fungi. They desperately needed an upgrade.
So here is our map for today's explainer. We'll start with the five-kingdom solution, move on to Monera, the survivors, then Protista, the misfits, followed by fungi, the decomposers, and wrap up with the future of classification.
Let's jump into Section 1, the five-kingdom solution. In 1969, R.H. Whittaker realized he had to look way beyond just plants versus animals to solve this sorting problem.
What you're looking at right here is your master cheat sheet. Whittaker built this brilliant matrix to logically place absolutely every living thing into a specific kingdom: Monera, Protista, fungi, Plantae, or Animalia. And he did it by asking five fundamental questions. What is the cell type? What's the cell wall made of? Is there a nuclear membrane? How is the body organized, and how does it get its nutrition? By sticking to these exact criteria, organisms that used to be incorrectly squished together, like, say, bacteria and blue-green algae being grouped with complex plants, were finally given their own distinct, proper categories.
Now, before we jump into our first specific kingdom, you absolutely need to memorize this crucial vocabulary trick. If a cell is prokaryotic, its genetic material isn't trapped inside a nuclear membrane. It's just kind of hanging out freely in the cell. So if you can remember that prokaryotic means no defined nucleus, you have already mastered the core defining feature of our first group.
Which brings us to Section 2, Kingdom Monera. These guys are famously known as the ultimate survivors of the microscopic world.
The primary rule of thumb for Monera is incredibly simple. If it's a single-celled prokaryote, boom, it stops right here. The sole members of this kingdom are bacteria, and they are literally everywhere. We're talking hundreds of them in a single handful of dirt. But what makes Monera truly fascinating is their survival skills. You've got archaebacteria surviving in boiling hot springs or wildly salty environments. Then you have eubacteria, which includes the stuff making your morning curd or fixing nitrogen in plant roots. Despite looking so simple, as a group, they actually possess the most extensive metabolic diversity of any organisms on Earth. It's wild.
Now, since thousands of bacteria exist, scientists use a fantastic visual trick to classify them without getting a headache. You just look at their shape. Use these four straightforward identifying terms. If it looks like a little sphere, it's a coccus. If it looks like a rod, it's a bacillus. If it's comma-shaped, it's a vibrio. And if it's a spiral, it's a spirillum. Memorize those four shapes, and you can instantly identify the broad category of almost any bacteria you see under a microscope.
Moving on to Section 3, Kingdom Protista. Let's step up the complexity ladder from those simple bacteria to a wildly diverse group that basically acts as the missing link between plants, animals, and fungi.
The absolute easiest trick to understanding Protista is to think of it as the ultimate catch-all or misfit category. Here is your checklist. Are they single-celled? Yes. Are they eukaryotic, meaning they finally have a true defined nucleus? Yes. Do they live primarily in water? Usually, yeah. Because this kingdom forms a link with plants, animals, and fungi, its boundaries are, frankly, not well defined at all. Basically, if it's a single-celled organism with a true nucleus and it doesn't neatly fit anywhere else, it gets tossed in here. And this visual brilliantly illustrates just how poorly defined this kingdom is. On one side, you have a euglena. In the sunlight, it acts like a plant, using photosynthesis. But take away the light, it becomes a heterotroph, literally hunting other tiny organisms. Then, on the other side, you have a paramecium, an actively moving predator covered in thousands of tiny hair-like cilia that literally sweep food into its gullet. They behave completely differently, yet they both belong to Protista. So let's rapidly break down these protista misfits with a simple list. You've got cruciferates, which include diatoms with cell walls embedded with silica, making them virtually indestructible. You have the frankly terrifying dinoflagellates. You have euglenoids, which lack a cell wall but have a super flexible protein layer instead. You've got slime molds, creeping along decaying leaves and engulfing organic material. And finally, protozoans, which act as primitive relatives of animals, living as predators or parasites.
Just to give you an idea of how dramatic these tiny misfits can be, check out this dinoflagellate. It uses stiff cellulose plates, kind of like biological armor, to protect itself. And some red dinoflagellates can multiply so incredibly fast that they create massive red tides, where the ocean literally appears red. The toxins they release during those blooms are so potent they can wipe out huge numbers of marine fish.
All right, before we transition to our next kingdom, you need to grab this specific trick word for your cheat sheet: dikaryon. During the sexual reproduction of certain fungi, there is this unique, temporary phase where a single cell has two completely distinct nuclei just hanging out together, in N+N phase, before they eventually fuse. Remembering this dikaryon phase is a massive shortcut to understanding fungal biology.
And that brings us to Section 4, Kingdom Fungi. These are the cosmopolitan organisms that might spoil your moist bread by turning it green, but they're also the exact same kingdom that provides us with life-saving antibiotics.
The crucial trick for identifying a fungus is its cell wall. Unlike plants, which use cellulose, fungi build their cell walls using a super tough substance called chitin. Furthermore, fungi are heterotrophic. They don't make their own food. Instead, they act as saprophytes, absorbing soluble organic matter from dead stuff. Their bodies mostly consist of long, thread-like structures called hyphae, which weave together to form a network known as a mycelium. Oh, and if you've ever wondered why we put food in the fridge, it's because fungi heavily prefer warm, humid environments.
Fungi are neatly divided into four main classes, primarily based on the morphology of their mycelium network and exactly how they form their spores and fruiting bodies. First, we have phycomycetes, which love damp wood. Then ascomycetes, or sac fungi, which includes everything from single-celled yeast to highly prized edible truffles. Next is basidiomycetes, which includes the common mushrooms you might eat, as well as destructive plant parasites like rusts. And finally, deuteromycetes.
Actually, there is a really fun trick to remember the deuteromycetes class, and it's their nickname, the imperfect fungi. But here's the secret: they aren't actually flawed. They are called imperfect simply because scientists haven't discovered their sexual reproduction phase yet. Once a researcher finally observes the sexual stage of a deuteromycete, it almost always gets immediately reclassified and moved into the ascomycetes or basidiomycetes class. It's basically the scientific waiting room for fungi.
Finally, let's look at Section 5, the future of classification. Because as robust as Whittaker's brilliant five-kingdom matrix is, this biological cheat sheet isn't perfectly set in stone forever.
The underlying takeaway you need to remember here is that biological classification is a living, breathing science. As our understanding of physiological and, more importantly, phylogenetic or evolutionary relationships improves, the rules actually adapt. For example, a newer three-domain system has already been proposed that takes our very first group, Kingdom Monera, and splits the bacteria into two entirely separate domains, resulting in a six-kingdom classification system.
Which leaves us with a pretty fascinating question to ponder. We have gone from Aristotle looking at red blood, to Linnaeus looking at just plants and animals, to Whittaker's five kingdoms, and now to three domains. So, if we discover a truly bizarre alien life form tomorrow, deep in an oceanic trench or out in space, will Whittaker's five-kingdom trick still hold up, or will we need to rewrite the biological cheat sheet entirely? Keep that in mind the next time you see a mushroom or a bacterial diagram. Thanks so much for joining me on this explainer.